Thank you very much, Charles. As I do my introduction, you can see my disclosures, which are in blue in this title slide. I'm going to be talking about resistance to checkpoint inhibitor immunotherapy because we have Carl June talking about CAR therapies later, so I won't talk about adoptive cell transfer therapy. And as opposed to what happens with targeted therapies, the main resistance problems with immunotherapy is the patients who don't respond up front. The patients who we give the therapy don't respond, as opposed to the patients who respond and develop resistance afterwards, which happens, but it's a minority of patients. And the patients who respond are like patients like this. These are two melanomas in sun-exposed areas with a lot of mutations, cancers that we had no treatment for. And if we block one negative regulator of the immune system, we can get a change from the left to the right. And if we see these patients, it's amazing because it continues to amaze me. I've been using this for several years. The T cells go in and start attacking the cancer. You don't even see inflammation around there. The T cells know what they attack. So in all of this immunotherapy, the T cell is the drug. Whatever we give to the patient is something that turns on the real killer, which is the T cell. And these ones follow Jim Allison's hypothesis of taking away a break to the immune system or immune checkpoint blockade therapy, for which he received the Nobel Prize earlier this year. And we know why this works. It's because these tumors had pre-existing T cells in there that were turned off by that checkpoint. And the T cells are stained in brown here. So you see very few at the beginning, and we block what's limiting them, and now we see an army of them. And this is not just one anecdote. This happens reproducibly. Here we looked at 22 cases, and we saw that the ones that had pre-existing T cells, the brown staining up here, were able to amplify this response over time with serial biopsies. And it's because these tumors had this interaction between the receptor, which is called PD-1, which is in green, and the ligand, which is PD-L1, which is in red. And actually, the presence of the T cell triggered the presence of the ligand that turned off that T cell. And the patients who don't respond tend to be like these 24 patients here, where we barely see any T cells at baseline or with therapy. PD-1 and CTLA-4 are co-receptors to the immune system. They only work if the main receptor is triggered. That's what gives the specificity for the cancer. And the main receptor is called the T cell receptor, which is an antibody-like molecule that's on the surface of the T cell that allows to signal once it's engaged by its cognate antigen, which is an MHC molecule that goes from inside the cell, takes some normal proteins and presents them on the surface. Nature developed that for us to fight bacteria and viruses. It's not that good to fight cancers that grow from inside because there's a series of negative regulators of the immune system. Every time we try to turn on the immune system has checks and balances, hopefully like the government, to turn itself off. Here we have the positive signaling, but the same signal leads to the expression of the PD-1 receptor. And that turns off this immune response. And now we have a T cell that tries to attack the cancer, did everything right, everything worked on activating the immune system, it made it to the tumor, but now the cancer protects itself, it senses it's under attack and stops that attack just by this interaction. So when that happens, we just give an antibody that blocks this negative interaction, and then we now can have a positive feedback loop in this interaction between the T cell and the tumor, where the tumor has receptors for a particular protein called interferon gamma that signals downstream. The negative regulator is taken out, and now we have all of the positive effects. Interferon gamma amplifies the response by increasing the antigen presentation. It attracts all the T cells, so it amplifies the signal every time it finds its antigen, and it has direct antiproliferative effects. That has led to this incredible change in how we treat cancer. And you saw Laura Glimcher talk this morning about this, so I'm just going to quickly reiterate. The first checkpoint inhibitor approved was ipilimumab in melanoma, which has activity, but it's only in this cancer. But here we have all of these other approvals in red dots by the Food and Drug Administration and many regulatories around the world. There's been 6 agents approved in 11 cancer indications in just 4 years, with this class of drugs that block PD-1 or PD-L1, which led to the second Nobel Prize earlier this month to Tasuku Honjo, and work by many others, as Laurie Glincher said. These therapies work because of these interactions, but by looking at the patients who responded and progressed, develop acquired resistance, we can start seeing why sometimes this doesn't work, even if the tumor is highly immunogenic. And we took a page from Charles Sawyer's book of studying acquired resistance and compared before and after. And then we can start seeing the genes that had changed afterwards in the patients who progressed. And if you look at this cartoon, you would say, well, if I was a cancer, I would try to get rid of this or this, because those are the main interaction points. So we get rid of the antigen presentation. There's nothing for the T-cell receptor to trigger, and that would not work. We found this in patients with primary and acquired resistance to melanoma and lung cancer in different papers that I'm reporting there, and also with model systems with CRISPR techniques to have brought to sample these mechanistically in preclinical models, found the same kind of losses. The other one, it's also obvious to anyone who's looking at this cartoon, which would be, what if you don't signal through interferon pathway? Then there's not going to be the amplification step, and then a T cell will not be able to be triggering all of these responses. And we've seen this with the primary and acquired resistance in patients with melanoma, and also the CRISPR screens also pulled this up. So one of the things that comes from is that we want antigen presentation, and we want the amplification, so triggering interferon response is important, which leads me to my last slide of how I be precision cancer immunotherapy, where you have a patient with a metastatic cancer, or earlier on, now we're starting to test in the adjuvant setting, and look in the tumor and see, is this biology there? Are there T cells in blue that are trying to attack the cancer, which is in brown, that are blocked by reactive expression of PD-L1? And if that's the case, all we need to do is to release that brake. Those T cells were there, ready to go. Everything else was working. That's in a minority of cancers throughout. Even in the ones where there's an approval, the majority of patients who get these agents will not respond to the therapy simply because this was not happening already. So in the ones where it's not happening, then we either have to bring T cells into the tumor or create the T cells. We can bring T cells into the tumor, maybe blocking two checkpoints. For example, CTLA-4 is an earlier checkpoint at the activation step. We block CTLA-4 and PD-1, which is an effector checkpoint, then we can get better responses. We can give immune-activating antibodies or cytokines that step on the gas at the same time you take away the brake. We use this symbol. We can use something like a modern iteration of choline toxins that Laura Glincher talked about this morning, TLR agonists on oncolytic viruses, to change the tumor microenvironment locally and then release its brakes. We can take immune suppressive cells, or we can combine with standard of care therapies, chemotherapies, and targeted therapies. Or finally, we can try to create these cells either by vaccination or genetic modification, which Carl June will talk to us later. Thank you very much.